Hi, and welcome to this lecture on environmental adaptation of animals. This is the last video on our famous omics section, and it's not on the nucleotides, but now on proteins and metabolism. Why on earth we should study proteins in this level? Well, because we have the nucleotide-based methods that are very efficient and quite cheap. So we can measure the activity of a single gene with real-time PCR, or we can check the activity of 1,000 genes with these gene chips. They are fast way to make science, but sometimes they're not giving you the right answer or not the whole truth. And we already know why it's so. We had this physical exercise. After half an hour, there was one type of genes that were upregulated. Some other genes were upregulated 4 hours later. So depending on what time you will take your sample, it's affecting on what kind of messenger RNA profile you will get. Then, besides this RNA processing, also the protein modification and translocation can affect on the amount of the protein or the protein activity. And this can cause you discrepancies that are pain in the ass in every research group. So you will get nice experiment done on tissue level, then nice experiments that are giving you same result in the cell level. Then you assume that, okay, this protein is upregulated or downregulated, and there's no change at all. It most probably is that, okay, you have detected the messenger RNA at the wrong time. But how to decide when to take the experiment? Should you take every day or every hour or every week? Well, that affects that, okay, more data points you need, more money you need. And then, of course, more animals you may also need. So it might be affecting so drastically that actually you must have some kind of compromise that, okay, you will take this, and then, but then the problem can be that if the data is not the same, giving us the same answer as in the protein level or in the activity level. Well, what you can do with the proteomics, you can study all the proteins, the number of all the proteins in the animal, when they are facing different kind of environmental adaptation, like adapting in the high altitudes. So we have humans living in the high altitudes, we have humans that are living on the sea level, and when taking a sample and isolating every single protein, we can say that, okay, is there more of some proteins on the humans that are living in the high altitudes. And when you are isolating these different proteins, you get a very messy picture. This is a two-dimensional gel where on the top there are the proteins that are heavy, lot of amino acids. Then in the bottom there are those proteins that are light, that are shorter ones. But because we can have at the same size proteins, we can have several different proteins. So that's why we need another dimensional. And because the proteins are formed by different amino acids, and different amino acids are behaving differently in different pH. So that's why we can have a pH axis to isolate different proteins. And that's why then each spot over here present a unique protein. And then when you are comparing several of these pictures together, then you find that, okay, in some pictures this spot is larger and over here it's smaller, etc. And then you can find some proteins in which the concentration is higher in high altitude or in the low altitude people. And quite often in this kind of experiment, some of these proteins are very obvious, like energy metabolism or oxygen binding proteins. But in some cases, there are some proteins that you definitely did not predict at all. Well, let's have another example from the insect world. We have an insect that is having a parasite. It's a parasite hairworm larva that is maturing inside this insect and then affecting on the proteins that exist in the brains of this insect. And it's the change in the number of proteins is affecting on the behavior of this insect. Because normally this insect is not trying to find water, but now it does. It seeks and enters the water, and when it's inside the water, then this parasite is exiting and breeding and producing larvae. So that's the life cycle of this parasite. And this can be compared quite easily by on the time when these animals are trying to find the water. So compare all the proteins of the insect that are looking for the water, and then to compare all the proteins from those animals also that are not looking the water. And over here, it's actually having several of these two-dimensional genes overlaid to see that some are increased during the water seeking and some are decreased. And actually, you can see that there are not so many dots in orange or green. So not so many proteins are affected on by this parasite. Well, beside the proteomics, we have also this metabolomics. And this is nothing to do with proteins or DNA, but it's not now on all kind of organic compounds in the animal. And so if we isolate all the organic compounds to see, and then we can see that, okay, is there more of these amino acids or more of these fatty acids, etc. And the way this kind of thing can be done, these methods are very chemical. So, for example, nuclear magnetic resonance can be used to identify that all different groups of these molecules, they can be identified with these peaks, and then compared that, for example, during the heat stress in the normal houseflies, they increase the tyrosine concentration, decrease the fatty acid amount of fatty acids inside these animals, etc. So they can have completely different kind of metabolism. Thank you.